All right, I hope everybody has had enjoyed their dinner and are enjoying dessert and coffee. It's truly my honor to introduce our two final speakers of this symposium. These really are two very special speakers. If you were there this morning, you heard that among the things that drove Professor Rosenberg was to help people. And of course, before you can help people, you have to help somebody. And we are very fortunate to have John Cleland here, who was the first patient cured by cisplatin. Likewise, of course, I think we heard time and time again that it really takes a team, it takes collaboration. And every member of that team is important. Some, of course, are absolutely critical. In this case, a person that was undoubtedly critical was our first speaker, actually. That's Dr. Larry Einhorn. Dr. Einhorn was the clinician who was treating, among others, John Cleland. Dr. Einhorn came to Indiana University in 1973, where he was, I think, the first medical oncologist there. At that time, in the early 70s, testicular cancer was the leading cause of death among young men. At the time, Dr. Einhorn joined IU. Very shortly thereafter, he began working and looking at cisplatin as a possible anti-cancer treatment. And it was an absolute critical time for cisplatin, critical time for those young men as well. And part of the reason it was a critical time was because at that point in time, due to some of the toxicity issues that we heard about today, there was resistance to continuing on and pursuing cisplatin as an anti-cancer drug. But it was Dr. Einhorn's data that continued to be the driver for what ultimately has become, again, a drug that has impacted millions of people, as well as we've talked about, has far-reaching consequences, even beyond the medical community. And so I know I am eager to hear from Dr. Einhorn, so I'll stop my introduction here and welcome to the podium Dr. Larry Einhorn.
Get unwound over here. Well, I hope you are enjoying your dessert, even though I'm not over here. But as long as you're happy, that's all I care about. So it's difficult after all day of listening to incredible talks, and they really were fantastic, and having a pretty heavy dinner. If anyone wants to close your eyes and maybe fall asleep a little bit, don't worry. You don't have a sleep disorder if that happens. But if I, as a speaker, start falling asleep, that's a real sleep disorder. So I'll try not to let that happen. So what I want to go over is what we've done as a clinical community as far as cisplatin. Oh, I'm sorry. Thank you. I'm just a rookie here. What do I know? So I want to go over what we've done with platinum and really where we've been in the past, present, and the future as far as platinum chemotherapy is concerned. And this is one of the true oncological success stories, probably more dramatic than any other drug as a single agent that's been used to combat cancer. So this is a structure that's been shown before, and the history of platinum is well known to this audience, and it first entered human clinical trials in 1972. I was a fellow at MD Anderson in 1973, and we used cisplatin on our developmental therapeutics program with Emil J. Freireich and Emil Frey in a variety of different tumors. And when I was a fellow, I was impressed with the horrendous toxicity, and we never really saw any patients with testicular cancer at MD Anderson who were treated with cisplatin during my year of fellowship there. And so when I came to Indiana University in 1973, cisplatin seemed to be an overly toxic drug with a hint of activity in multiple different tumors. And certainly the early toxicity outweighed therapeutic advantage. But if a drug works, as a scientific community, we figure out how to combat and mitigate that type of toxicity, and that's what I'm going to focus on the early part of this lecture. So this is the type of toxicity that is seen with cisplatin. So anorexia, nausea, and vomiting was really bad, and let me explain what really bad is. Cisplatin is given for 5 days in a row. On day 1 of that 5 days, the average number of emetic episodes was 12. 12. That's day 1 of a 5-day course. Obviously, anorexia is associated with the nausea and vomiting. For those patients in the early clinical trials with diseases other than testis cancer, if they responded and got a second course 3 weeks later, a third course 3 weeks later, a fourth course 3 weeks later, they would develop acute renal failure. This should not have been a surprise, but it was. We were used to chemotherapy causing hair loss, causing sores in the mouth, causing nausea and vomiting. We were not used to drugs that actually was affecting organs, such as nephrotoxicity with cisplatin. It became relatively simple, and it wasn't rocket science to figure out how to combat the nephrotoxicity, because other heavy metals, like mercury, were known to cause acute proximal tubular necrosis. And that can be ameliorated by simply hydrating a patient before they get a heavy metal, and then to ensure that the drug doesn't accumulate in the proximal tubules by giving simple saline hydration. So simple pre-hydration and post-hydration solve the problem of nephrotoxicity. Neurotoxicity, as you heard this afternoon, is still a problem. This is the same neurotoxicity that diabetics get. It's called stocking glove paresthesias. Numbness and tingling in the hands and feet. Sorry.
So getting some feedback from your mic, so if you could maybe hold it a little bit further away or put it up there.
Okay, thank you. How's this? Does it work okay? Okay, thank you, sorry. So neurotoxicity is a cumulative side effect of heavy metal. Lead is another heavy metal that causes peripheral neuropathy. So we recognize the axonal degeneration that's seen with cisplatin-based neurotoxicity. We have an NCI think tank coming up in September looking at neurotoxicity 44 years after cisplatin was first introduced into clinical trials. Obviously, we haven't solved that problem at the present time. Now, ototoxicity, as you heard this morning, the drug accumulates in the cochlear areas and the organ of corti, and ototoxicity, which is manifested with ringing in the ears and high-frequency hearing loss. So young men with testicular cancer had a rationale for going like this when their wife would be telling them something, because it was real. They really could not hear their wives. They had high-frequency hearing loss. So in the New England Journal of Medicine just last month was some hope for patients with ototoxicity in a pediatric disease, which is a rare disease, hepatoblastoma. So the use of an old drug, sodium thiosulfate, seemed to greatly mitigate the ototoxicity that was seen in very small, very young children. And whether that will be true for adults remains to be seen. This drug also lowers the calcium, lowers the magnesium, causes some degree of anemia, and sterility. So platinum affects the Leydig cells in the testis that make testosterone, and it also affects fertility. So when we cure a young man with testicular cancer, we know that 40% of the time, that person will have permanent azospermia or oligospermia and be unable to father a child. And this is, of course, the reason why we do sperm banking prior to starting cisplatin-based chemotherapy on young men who have testicular cancer. So these are some of the unique characteristics of platinum. In addition to its broad spectrum of activity, which I'll get back to later on, platinum, unlike any chemotherapy drug, is synergistic across a wide panoply of a variety of different chemotherapeutic agents. The most dramatic is 5-fluorouracil, fluorinated pyrimidines, etoposide, many different drugs. Platinum plus that drug is not 1 + 1 = 2, but 1 + 1 = 3, with dramatic synergism. There is no other drug that has that like platinum does. Also, platinum, we use the term combined modality therapy in many diseases like lung cancer, cervical cancer, esophageal cancer, head and neck cancer. There are no drugs that combine as well with radiotherapy and synergize the effectiveness of local control with radiotherapy than cisplatin. So even if this drug did not have that broad spectrum of activity, It is unique among all chemotherapeutic agents based upon its synergism and its being able to be combined with radiation therapy. And these are, again, the drugs that platinum is truly synergistic with. So testicular cancer. So testicular cancer accounts for just 1% of all male cancer. Only 1 out of 400 men get testicular cancer during their lifetime. The only known risk factor is cryptorchidism, being born with undescended testes. But 90% of our patients that have testis cancer have no history of cryptorchidism. So its etiology remains elusive. We also don't know why this is a young man's disease. Most cancer is a disease of aging. It's a genetic disease, not because you inherit cancer from your parents, but all it takes is one mutation in one gene to cause the cascade of events that causes phenotypical cancer. So nevertheless, despite the paucity of cases, this is the most common carcinoma in young men. So what's the most common male cancer? Prostate cancer. And not that it's not tragic if someone dies at age 80 from prostate cancer. If someone dies from testis cancer, they literally are losing 50 years of productive life lost. Now, this is also an important disease because the goal of therapy is not palliation, nor is it prolongation of survival. The goal of therapy is cure, and fortunately, we're able to achieve that goal in the great majority of cases. We also can do studies which have been done by our pediatric colleagues in acute lymphoblastic leukemia and other childhood malignancies, because all patients who get chemotherapy for testis cancer have platinum-based chemotherapy, and we can look at some of the late consequences of therapy because fortunately we have patients that we can follow for 10, 20, 30, 40 plus years. Well, before there was platinum, there was actinomycin D, a drug, by the way, that caused just as severe nausea and vomiting as platinum did. And in the 1960s and early 1970s, we actually saw some cures with actinomycin D. And for non-hematological malignancies, non-lymphomas, non-leukemias, the fact that we can cure a solid tumor was pretty remarkable, and this was done with actinomycin D. Now, when I was at MD Anderson, Dr. Mel Samuels was the first to look at the single-agent activity of two other drugs, vinblastine and bleomycin. They were neither better or worse than actinomycin D as far as efficacy is concerned, but that two-drug combination was also synergistic, achieving a 25% cure rate, much higher than you would expect by combining two drugs that have a 5% cure rate. But obviously, what revolutionized the cure in this disease was Barney Rosenberg's discovery of platinum. When platinum was done as a phase 1 trial when I was at MD Anderson, again, as I mentioned, to the best of my knowledge, we didn't have any patients with testis cancer treated during my fellowship with that particular drug. So the first study came out of Roswell Park in Buffalo, New York, and they treated 11 patients in their phase 1 series. They had a lot of colon cancer, lung cancer, prostate cancer, the usual type of diseases that are common malignancies. So out of those 11 patients, 3 complete and 3 partial remissions. Now, when you're using an experimental drug, if you see a hint of clinical activity in heavily pretreated patients, you get very excited. But to have complete disappearance of pulmonary metastases, really then and now is pretty well unheard of with a chemotherapy drug. However, this was third-line or fourth-line therapy as a single agent. None of those patients were cured with single-agent cisplatin. But it set the tone for looking at platinum as a component of a combination chemotherapy. Diseases that we cure, like lymphomas, childhood leukemia, and of course testicular cancer, are not cured with single-agent therapy. They're cured by combining drugs that have known single-agent activity, that have different mechanisms of cytotoxicity, that do not have overlapping toxicity. The major dose-limiting toxicity for chemotherapy is myelosuppression, and are synergistic, all of which were true with these 3 drugs, and the only myelosuppressive drug in the combination was vinblastine. Now, when we did this first phase 2 study, if there was a model for a curable neoplasm, It was childhood acute lymphoblastic leukemia, with the elegant studies that were done at St. Jude's Hospital curing the majority of children who are 3, 4, 5 years old with acute leukemia. In that model, it was felt that you needed to give induction therapy followed by 2 years of maintenance therapy. So that was the mantra in all of oncology in the 1970s, and ergo, after giving cisplatin and bleomycin for the 4 courses of therapy, they then received maintenance single-agent vinblastine for a total of 2 years. So again, these are the mechanisms for combination chemotherapy. Now, I would admit that even in my youthful exuberance, I never would have dreamed that the introduction of a single experimental drug, cisplatin, would so have changed the face. We literally had a 1 logarithmic increase in the cure rate with the number of patients currently showing no evidence of disease compared to contemporaneous actinomycin D studies with its 5% cure rate. Well, if something works in Indianapolis, there should be technology transfer, and it will work anywhere in the United States and outside of the United States. So this is SEER data that came out of the NCI after our first abstract was presented at our national meeting and our first paper was presented, showing that the cure rate, its survival rate, with the introduction of platinum had this upward movement that we've never before, and quite honestly, never since have seen in any individual cancer due to the dramatic results with testicular cancer. Well, to show you a couple of the before-after pictures back in the early 1970s, this is a patient who came to us critically ill, severely hypoxic, and in fact, on his lateral exam, there's almost no black on that chest x-ray. All that white is tumor. If this were a disease, anything other than testis cancer, we would not have treated this patient. We would have sent him straight to hospice. But this is testis cancer, and he's long since been cured. This is another one of our patients with choriocarcinoma, with massive pulmonary metastases, again with everything disappearing. And this is liver metastases. All those round black spots in the liver, of course, represent metastatic embryonal carcinoma in this particular case, again with complete disappearance of disease. And those little tiny things that you see are just scar tissue with remnants of the massive disease that they had before. So this is where we were in 1976 when we finished our phase 2 study with cisplatin, vinblastine, and bleomycin. We then did a variety of phase 3 studies because by 1976 we had convinced at least ourselves that these spectacular results had nothing to do with the 2 older drugs, but were primarily due to cisplatin. So the first phase 3 study we did is we reduced the dosage of vinblastine that lessened the myelosuppression and lessened the neuromuscular toxicity associated with that particular drug. And there was no difference whether we gave the older dosage or the higher dose or the 25% dosage reduction. The next phase 3 study we did, we challenged one of the basic tenets in oncology about long-term maintenance therapy. So we randomized patients who achieved a complete remission to receive or not to receive those additional 21 months of vinblastine. And we were gratified that there was not even a 1% difference in cure rate. So we eliminated the high toxicity with vinblastine from a phase 3 study, and we eliminated the 21 months of maintenance therapy. Well, synergism is very easy to prove in a mouse. And this is data from Skipper and Chabrol from P388 leukemia. And this shows that when you inject tumor cells into the ventral surface of a mouse, it's very easy to tell if a drug works and if 2 drugs are synergistic. So if nothing is done, the mice all die very quickly. Single agent cisplatin in this mouse model prolongs survival very minimally. This is a newer drug in the early 1980s, etoposide or VP16, which again had a couple week prolongation of survival. But when the 2 drugs were combined in this mouse model, there was a 25% murine cure rate. So this is a nice preclinical model of synergism that came from Skipper and Chabrol's lab in Southern Research Institute in Alabama. So with that as a background, we looked at this as second-line chemotherapy for patients who were not cured with cisplatin, vinblastine bleomycin. So we combined these two drugs, and much like in the animal model, we cured about 25% of our patients. This was the first time that an adult solid tumor had ever been cured with a form of second-line chemotherapy. So with that as a background, our next phase 3 study, we wanted to see whether we can substitute vinblastine for this newer drug, etoposide, or VP16. And before the first patient went on this phase 3 study, we knew that etoposide had no neuromuscular toxicity and that this was going to be a less toxic regimen, but would it be an equivalent regimen? Well, not only was it equivalent, but it was superior. So the number of men with continuous no evidence of disease with the BEP regimen was 12% higher. So when this study finished in 1984, we now relegated the original PVV regimen to historical footnote only based upon this large phase 3 study demonstrating less toxicity and clinically superior efficacy with the BEP regimen. Well, we also looked at duration of therapy. So during these time periods, we were beginning to understand a little bit about platinum-induced nausea and vomiting. Now, when someone gets sick from chemotherapy, unlike what patients often think, it's not because they have a weak stomach. It has to do with a central nervous system effect. So there's a chemoreceptor trigger zone, and there's an emetic center in the medulla oblongata. And it's the drugs that affect those central nervous systems that actually causes chemotherapy-induced nausea and vomiting. Now, basic scientists use animal models by injecting apomorphine, which kind of mimics some of the things that happen with chemotherapy. And the animal model that has been used is the ferret, and I hate to say this, the poor little pussycat. So it's very easy to study things in that type of model. And one of the things that was discovered in those models and also in humans also is when platinum is injected, moments later, a chemical, 5-hydroxytryptamine 3 or 5-HT3 is secreted. So it then became a logical thing to look at developing drugs that inhibit 5-HT3. And the first of these was ondansetron. So when ondansetron came along, instead of an average number of emetic episodes of 12 on day 1, it became 0 to 1. Just as cisplatin was such a dramatic drug, so was ondansetron. It allowed us to give everyone therapy as an outpatient rather than giving continuous IV hydration for 5 days, not just because of the severe nausea and vomiting, but people get very dehydrated with nausea and vomiting. They then get pre-renal azotemia and go into acute renal failure together with the cisplatin. So the study that we then did is we looked at 4 courses of BEP compared to 3 courses of BEP in those patients that didn't have such massive metastatic disease. And once again, we didn't even see a 1% difference. So over this 13-year period of time, we went from everyone having 2 years of chemotherapy to substituting etoposide for vinblastine and showing less toxicity and higher cure rate, and allowing us to cure patients with 9 weeks of chemotherapy as opposed to the original 2 years, or in these studies, the 12 weeks of chemotherapy. Well, what do we do today when we don't cure somebody with platinum-based chemotherapy, be it with BEP chemotherapy or similar type of chemotherapy? Well, most of the lay public is familiar with the term bone marrow transplant. And there are 2 types of bone marrow transplant. What we use here is autologous transplant, where we are taking the patient's own healthy hematopoietic stem cells so that we can give someone 5 times the dosage of a myelosuppressive drug, and then we reinfuse a patient's own healthy stem cells. So that is not therapeutic transplant. The transplant simply allows us to give higher dosages of myelosuppressive therapy. On the leukemia service, we do allogeneic transplant, where we take an HOA-compatible sibling, and we can actually get a graft versus tumor response, as well as allowing us to get such high dosages of chemotherapy. Well, in the 1980s, the most commonly treated disease with high-dose chemotherapy and bone marrow transplant was breast cancer. and it was a totally failed experiment. And it was a matter of investigators having optimism overcome reality. And they looked at a series of patients that were treated with high-dose chemotherapy and bone marrow transplant and compared it to historical controls. And anytime you compare it to historical controls, you're comparing it to a different decade, different technology for assessing disease. And all those studies that looked so positive, when they were done as phase 3 studies, every one of those studies in North America, in Europe, every one of them were negative. So it really cast a pallor on the idea of doing high-dose chemotherapy. But it's easy to understand why it didn't work in breast cancer. Again, the transplant is not therapy. It's supportive care to allow you to give higher dosages of myelosuppressive agents. What are the best drugs in breast cancer? Doxorubicin, which is adriamycin, which can cause cardiac damage if you give it in high dose, and the taxanes, paclitaxel and docetaxel, that will cause severe neuropathy and even encephalopathy in high dosages. So you can't give those drugs in high dosages. What's the best drug in testis cancer? Cisplatin, obviously. If we give 5 times the dose of cisplatin, the patients would be on dialysis permanently or being renal transplant patients. They would be crippled with neuropathy and they would be deaf. And that's where Hillary's studies were so important with the use of how to properly use carboplatin. So carboplatin's main toxicity, as Dr. Calvert mentioned, is thrombocytopenia. So that's something we can combat when we're giving patients healthy hematopoietic stem cells. So with the discovery of carboplatin in clinical trials in 1986, that's when we started our bone marrow transplant studies. What's the second best drug in testis cancer? Etoposide, VP16. Its only dose-limiting toxicity is myelosuppression. So in 1996, 10 years into doing bone marrow transplant, we switched to what's called peripheral blood stem cell transplant. Much easier to do. It allowed more rapid engraftment with patients recovering from the hematological complications before going on to a second course of high-dose chemotherapy. And Hillary, for your information, we did this on a milligram per meter squared basis with the carboplatin, as you can see. But when we translate that to your formula for AUC, the average AUC for these patients was 32. It's a hefty AUC. So what were the results? So in this first series that was reported in the New England Journal of Medicine, 184 consecutive patients, we cured almost 2/3 of these patients. Now, to me, it is truly remarkable what has been done with cisplatin with first-line therapy in curing disease. But it's a miracle that we can take the same drugs that the patient failed to be cured with and do great escalation with peripheral blood stem cell transplant and still be able to have this type of cure rate in this patient population. And we updated, well, I'm sorry, this is just one of the patients that we treated with a large retroperitoneal mass and liver metastases, as shown over here, and after the high-dose chemotherapy, complete disappearance of his disease. So looking back before platinum was around these ancient times, BC, before cisplatin, so this is looking at our various stages of disease. In any cancer, we stage according to the extent of disease. So stage 1 disease meant that disease was confined clinically to the testis, and many of these patients were cured with orchiectomy alone. Stage 2 disease meant that it had spread to the regional retroperitoneal lymph nodes in the abdomen, and urologists, by doing retroperitoneal lymph node dissections, were curing about half of the patients. And in the actinomycin D era, when you had pulmonary metastases, stage 3 disease, 90% of these patients died within the first year of therapy. And this is where we are today with the treatment of testis cancer. Almost everyone is initially or subsequently, when they relapse after surgery, cured with platinum-based chemotherapy. And for those patients that present with stage 3 disease, 80% of these patients are cured. So overall, we have a 95% cure rate in patients with testicular cancer. Well, times have changed as to how we treat cancer. And I agree completely with virtually every speaker who came up to the podium and saying that chemotherapy is not dead, platinum is not dead, this is still such a major drug in so many different diseases. But we are moving away from chemotherapy as we understand the biology of cancer itself. We are looking at what are the specific genotypic mutations that can allow a tumor to develop and to progress and proliferate. And we look at molecular targeted agents, and we've heard talks already about precision medicine. And what really has changed things is immunotherapy. Immunotherapy has been transformational in many diseases such as melanoma, where we are probably curing about 20% of patients with melanoma, with metastatic disease, and we are greatly prolonging survival in patients with lung cancer. But platinum is not going away, and platinum is still used in diseases like lung cancer, as I'll get to back later on. And this is a classic paper looking at the hallmarks of cancer. And these are some of the molecular targeted agents that we have available in 2018 that are making such a difference in how cancer is treated, as opposed to giving the same chemotherapy recipe to every single patient with that type of cancer based upon the organ of origination rather than the mutation that is driving the cancer itself. So nevertheless, despite the enthusiasm of the public and the scientific community for immunotherapy, for molecular targeted agents, platinum in 2018 is still first-line therapy for these 11 different diseases. And these abbreviations are small cell lung cancer and non-small cell lung cancer. I did not list breast cancer here because breast cancer is treated with first-line therapy with platinum in just a small cohort of patients, whereas platinum is first-line therapy in every one of these patients, often used as adjuvant therapy after surgery, improving the cure rate, often combined with radiotherapy in diseases like cervical cancer, esophageal cancer, and always used in metastatic disease. It is truly remarkable, 40 years after FDA approval, that this single drug is still first-line therapy and reflects curative intent, not just in testis cancer, but many of the other diseases that are listed here. And the final comment that I will make is that platinum has saved the lives of so many young men, thousands, tens of thousands, hundreds of thousands young men. But testis cancer arguably saved platinum, because were it not for the truly remarkable results that we're seeing with testicular cancer, it is questionable whether this drug would have been pursued and the efforts gone into it that went into it with Barney Rosenberg's wonderful discovery. So I believe that that is my last. Oh, I'm sorry, I have a couple more here. Sorry. You know, I changed a couple slides at the end over here. So looking at the future over here, as far as platinum is concerned, you know, I'm not sure if there will ever be another chemotherapy agent as effective as cisplatin. Certainly immunotherapy has a wide spectrum of activity as platinum does, but we have yet to prove, other than melanoma, that we're curing anybody with immunotherapy despite the dramatic responses that we are seeing. And this is a very famous book that many of you are familiar with, The Emperor of All Maladies. And this was written by Sid Mukherjee when he was an oncology fellow at Dana-Farber. And as you can read, it says, in the end, cancer truly emerges, as a 19th century surgeon wrote, run in a book's front piece, as the emperor of all maladies, the king of terrors. Well, it's no longer the emperor of all maladies. It's no longer the king of terrors. Barney Rosenberg had a lot to do with this. Cancer is still a formidable, fearsome disease, but it's not the formidable, fearsome disease that it was once before. So again, this is like a shrine for me coming here. Jim, I appreciate the invitation, and thank you very much.
Questions?
Sure.
So Dr. Einhorn would be happy to take any questions. Well, I actually have one. So
Okay.
from the time when Barney and Loretta made the observation, how actually did cisplatin get to you?
Sure. Well, you know, I asked one of the questions this morning whether Barney had any idea of how profound this drug would be. All of us that work in cancer research, we have our own babies and we think it's going to do a difference, but such an incredible difference. So the way cisplatin came to us, when I joined the faculty at Indiana University, I had finished a fellowship in developmental therapeutics at MD Anderson for 1 year. And so we were looking at a variety of experimental agents. As I mentioned, I had bad experience with cisplatin because we were just seeing the early toxicity and not the later therapeutic advances. And we came across an article that cisplatin causes testicular atrophy. I thought, gee, isn't that interesting? In my naive experience, I thought that might work for testis cancer before I realized that most chemotherapy drugs cause testicular atrophy. And then we saw the data from Roswell Park, single agent cisplatin, showing these type of responses but no cures. And so I felt that this was a new, exciting drug with a unique mechanism of action. It was a non-myelosuppressive drug, and we felt that this would be logical. And I mentioned to a couple of people at lunch that back then, things were much easier than they are in 2018 for an investigator. So I wrote up a protocol with PVB, platinum vinblastine bleomycin. We sent it to the NCI, they approved it, and they sent us the drugs. Well, the person who reviewed the protocol turned it down and said that if we use platinum as first-line therapy, what are you going to give patients as second-line therapy? I was not deterred, so I went to someone above him, and we were able to get the drug. So that's how we wound up getting cisplatin. It used to come in a big package with all the vials, you know, would just go to the post office and pick it up. Times were a little simpler then. And for the clinical people in the audience, we wrote a protocol, and within 3 weeks, we treated our first patient. Today, you write a protocol, if it's 18 months, you're lucky to get the disease treated. And John Cleland's going to be standing up here in just a few minutes, my good friend and my former patient. So John was the second patient that we treated with PVB. The first patient died of kidney failure. He was heavily pretreated, quite ill, but we didn't know about hydration at the time. If that happened today, we would have been shut down for a year before we would have been able to treat the next patient. So you don't want someone being a maverick doing crazy things out there, but times have changed as far as clinical research, and not necessarily all for the better. Steve?
Are any patients ever given platinum without any surgery first?
No, because when someone has testicular cancer, there are two areas where intravenous drugs don't have adequate access. The first is the brain, the so-called blood-brain barrier, but the other is the testis. So for unknown reasons, although it atrophies the testis and affects fertility, the drug does not get into the testis as well as the rest of the body, and we know that not by supposition, but by actual clinical observation. We would see patients that would come to us with huge abdominal masses, critically ill, with a mass in the testis, and the biopsy would show testicular cancer of the abdominal mass, and so we would treat them immediately before doing the surgery to remove the testis, and then we would resect the residual abdominal mass, which was all dead scar tissue, and we would resect this tiny little tumor in the testis, and about 20% of those in the testis still had active cancer in the testis. So that's how we learned about the testis being an active site. So even if you're giving someone an intravenous cytolytic agent, you still have to remove the testis because the drug doesn't get into the testis as well as it does other parts of the body.
So if there's somebody, I mean, I know of cases where people have been cured of testicular cancer, but only one testicle was taken and the other one was used and they had children.
Oh, absolutely. This is not a bilateral disease. So testis cancer spreads upward, like up an elevator shaft through the lymphatics to the retroperitoneum, mediastinum, cervical lymph nodes, or hematogenous to the lungs, and later to liver, bone, and brain as far as the sites of metastatic spread. So patients often ask us that same question, do I have to have my other testis removed? And the answer is no, we don't get transscrotal spread. But when someone is cured of testis cancer, they have a higher incidence of a second different primary, and that happens 1% of the time as opposed to 1 out of 400, which is a general male population. So some of our patients have the bad luck to have been struck twice and get a second testis cancer in the other testis. They wind up being on lifetime replacement testosterone. They're infertile when that happens. But our patients, sperm banking is done. We do, when we do a retroperitoneal lymph node dissection, we do a nerve-sparing procedure, so we're not cutting the sympathetic nerves that control forward movement of sperm. So the great majority of our patients, and you'll see John standing up here in a few minutes, or looking at Lance Armstrong or any other patients, really pretty much get back to their life full time. That yes, there is some bothersome neuropathy and ototoxicity, but these are relatively minor type of problems. There are some patients that, despite sperm banking, doesn't take, they're not able to father a child, and as I mentioned before, we tell our patients up front that 40% of the time they're going to still have oligospermia or azospermia, and that's not going to go away like the other symptoms of the acute toxicity will go away.
Yes, you sort of begged the question, will there be another new chemical class?
Well, yes, and you know, Steve gave such a great lecture, and one of the drugs that he mentioned, netaplatin. Netaplatin was just in Lancet Oncology, and it came out of Asia, but it looks very promising, not because of better activity, but because when used in head and neck cancer combined with radiotherapy, that's a tough treatment regimen of platinum plus concurrent radiotherapy with severe mucositis, severe nausea and vomiting. And so this drug maintained in a large phase 3 study the same therapeutic efficacy and had none of the type of toxicity that cisplatin did. So again, I think to me, the goal is not to have a less toxic platinum. The goal is to look at oxaliplatin works in colon cancer, cisplatin and carboplatin don't. So we want to be looking at new agents. And I love Steve's talk. It was phenomenal. And I think that there are ways of looking at why does platinum work so well, and can we get newer platinums, and can we do mechanisms to overcome resistance with the platinum? So I don't think that the future is looking at a less toxic platinum. I think the future is trying to look at a better understanding of why platinum works so well in testis cancer, and how can we use this knowledge for other tumors and develop other platinums. And unfortunately, and I mentioned this when I was with people at lunch, the major money that comes into drug development, as you all know, is not from the NCI, it's from industry. Industry has become weary of chemotherapy, and it's really too sad. I mean, everyone's into the immunotherapy and molecular targeted agents, and that's great, but it shouldn't be done with the exclusion of what can be accomplished with trying to develop a better platinum.
What happened, that first experiment on sarcoma 180, I was there.
Right.
That first experiment showed we didn't need statistics. We had cures.
Yeah.
First experiment. How many new agents do that?
Yeah, exactly.
That's the point.
Yeah.
That's the rarity of the discovery.
Yeah, no, absolutely. It was a phenomenal discovery, and when I was being interviewed during one of the talks I had of Miss, I made the comment that, and several others made the comment too, I think it's disgraceful that Barney never got the Nobel Prize. I think with the exception of Jonas Salk, I don't know anyone that has done more to cure human illness than what he did with platinum, and it's just remarkable. And it, you know, it's like applying for a grant or putting a manuscript to a journal. You know, the peer review process is not always perfect, and there's a lot of flaw in that, and it's just too bad it never happened, because it certainly should have. And as I said at the beginning of my talk, to me coming here, this is like a shrine because of what's been done at this great institution.
We'll take one more question.
If we have a appropriate biomarker, can we increase the sensitivity of the patient?
Well, we look at biomarkers with our molecular targeted agents, and the other area of my research is lung cancer. So lung cancer has changed dramatically, and we have 8 different molecular targets that we can look at and do personalized medicine. In testis cancer, to tell you the truth, we don't need a molecular target. The drug always works. In 40 years, I have yet to see a single patient who initially got platinum that didn't respond to it dramatically well. They're not all cured with it, but they respond to it dramatically. But biomarkers are important in all diseases, perhaps except testis cancer, because we don't need a biomarker.